Hi everyone, welcome to the latest installment of the HDSA research webinar series. This is George Orling from the National Offices of HDSA, and I'm really excited to have the latest installment, which is going to be a talk from Dr. Ottavio Vitolo. He'll be presenting an overview of the clinical study being sponsored by Pfizer called the Amarilla study. And a couple of years ago, we had a webinar in the same series on PDE10 and the work that was going on to investigate the levels of PDE10 as a potential biomarker for disease progression in HD. And today, what you'll hear is more specifics about this now interventional drug study being sponsored by Pfizer to test to see if the inhibition of PDE10 will exert any therapeutic or beneficial effects in Huntington's disease patients. So presenting today is Dr. Ottavio Vitolo. He's the Senior Director and Head of Clinical Neuromuscular Research in the Rare Disease Research Unit at Pfizer, he's based in Cambridge, Massachusetts. Ottavio is a neurophysicist with more than 15 years of research and clinical experience in the field of neurodegenerative disorders. He graduated from the School of Medicine at the University of Rome in La Sapienza, Italy, and trained in psychiatry at WashU in St. Louis, as well as in Brigham and Women's Hospital at Harvard Medical School. He's been on the faculty at the Department of Psychiatry at Mass General Hospital and Harvard Medical. And before joining Pfizer, he was working at Shire Human Genetics Therapy. So before I turn it over to Ottavio to tell you a little bit more about the Amarilla study, I just want to take a couple minutes to, for those who may be new to the webinar series, just to tell you how you can ask questions during the course of Ottavio's talk. So on the right-hand side of your screen, you should see a box, a menu, where you can go into the questions section, shown here in the red box, and type your question and click send at any time during the talk. And at the end of the presentation, we'll go through as many of the questions as possible to try to get them answered by Dr. Vitolo. We are recording this, and approximately within 1 week after this webinar, hopefully earlier, we will try our best to get this both on our national website as well as on our YouTube channel. So if you have to bail out before the end, or you want to share this information with family members or other HD patients that you may know might be interested, they'll be able to view this at their convenience. And I'll just take 1 minute to say that we have one more webinar scheduled for 2015, and on December 9th at 12 p.m. Eastern, Dr. Dawn Lowe from UCLA will be giving a presentation on a study being run out of UCLA on at-home monitoring of sleep-wake cycles in Huntington's disease patients. This is actually a study that's being sponsored by HDSA through our Human Biology Project. So please join us on December 9th for that exciting presentation. So with all that said, I'd like to turn it over now to Dr. Vitolo, who will talk you through the Amarilla study. Turn it over to you, and you can share your screen.
Thank you, George. Can you see my screen?
I can.
Okay, wonderful. Well, thanks so much again for inviting me to speak about the PDE10 program in Huntington disease that we have been conducting at Pfizer for quite a number of years now. Some of you in the audience may have already seen this presentation if you were at the HDSA meeting in Dallas in June of this year. I will provide, again, an overview of the program, speak about the Amarilla study in particular, but I will also give you a little update on a recently concluded study that was done in France, in Paris, that has provided us additional information on how this compound may work in Huntington's disease. So let's move ahead. I think I will remove. Do you see my right bar as well?
Oh, no, you're okay.
No, I'm okay. All right. Sorry about that. So I'd like to start looking at the brain changes and manifestations of Huntington's disease, because that's important to put the drug activity in context. Then I will review what the compound does to the brain, and then I will go over what effects we may expect from the compound in Huntington's disease. And finally, I will speak in more details about the Amarilla study, which is currently ongoing. So what are the brain changes and manifestations of Huntington's disease? Many of you are very well familiar with them, but I think it's good to have a little bit of a refresher and maybe explain to those of you who have not heard it before. So Huntington's disease is characterized by degeneration of a brain area called the striatum, which is here depicted in purple. And the striatum is a very important area of the brain because it helps connecting different parts of the brain and sorts out information in a certain way. So it's connected primarily with the cortex, which I'm trying to highlight here, and with deeper structures like the thalamus, which is here next to it. And what happens in Huntington's disease is that these cells, these neurons, so these nervous cells in the striatum, which are called the medium spiny neurons, due to their conformation, it looks like they have little tiny spines. They become impaired. They don't function as they should initially, and eventually they degenerate and die. And that's thought to be related to accumulation of aggregated mutated Huntington protein. And here in this little panel, you have an image at the microscope of these aggregates. So the degeneration of these cells basically cuts off the communication between the cortex and the striatum and other brain areas. And that's thought to be responsible for many of the clinical manifestations of the disease manifestations, like the well-known motor problems with chorea, the problems with voluntary control of movements, problems with coordination, but also the cognitive problems appear to be related to dysfunction of these circuitry, of this connection between the striatum and the cortex. And so problems like attention and concentration problems, loss of mental flexibility, deficit in mental planning are very likely related to this dysfunction of this brain circuitry. And also the behavioral problems typically seen in HD can be reconducted to these same mechanism. So what does the compound do to the brain? Why PD-10 and the PD-10 inhibitor, how it is called? So it turns out that these small neurons, these small cells in the striatum, have high levels of this enzyme. An enzyme is basically a protein that transforms other molecules, makes them either work better or stops them from being effectively working. And so PD-10 is highly expressed in these medium spiny neurons and helps the medium spiny neurons function. We know that mutated Huntington can decrease the levels of PD-10. I have to correct myself. Actually, can decrease the levels of two molecules controlled by PD-10, making them less available. So the idea is that inhibiting PD-10, we would be able to counteract the effect of the mutated Huntington on these two other molecules, which are called the cyclic AMP and cyclic GMP. And what do these molecules do? They actually transfer the information from the surface of the cells to the nucleus, to basically the major engine of the cell. And so the idea is that we are helping with our compound, the cells, functioning better, restoring basically these communication within the cell that is impaired by mutated Huntington. And ultimately, the result of this effect is improved communication between the cortex, that basically area of the brain that is on the surface, and the deep nuclei, the striatum, where the medium spiny neurons are. So it's not basically a cure for Huntington disease, but potentially could counteract the effects of mutated Huntington and improve function in Huntington disease. And so what are the effects that we may expect from this compound in Huntington disease? Well, as I mentioned before, the critical role of the striatum, of this area of the brain here in purple, defined by this purple circle, in controlling motor function, cognition, and behavior, gives us hope and makes us believe that the compound could improve the three major disease manifestations of Huntington disease. So it could improve, for example, chorea and other movements, could improve cognitive function, and it could improve behavior. And of course, this is for now based on primarily on animal data, but just recently we got some news from a completed study that make us believe that this is possible also in humans. So what is this Amarilla study? The Amarilla study is what was in jargon called a phase 2 study. So it's a study that tests the efficacy, so whether the compound really is supposed to do what we hope it does. In this case, if it improves the clinical manifestations of Huntington disease. And safety as well. We want to make sure that the compound actually is safe for people who have Huntington disease. It is a relatively large study. We expect to have 260 participants aged 30 to 65 who are in the early stages of HD. So the so-called HD stage 1 and 2. And these are studies conducted at 46 centers across the world in 5 different countries, and that includes Canada, the US, UK, Germany, and Poland. And people who enter this study are randomly assigned, so they're assigned by chance, basically, to 3 different arms, 3 different treatments. They can receive the placebo, or they can receive 2 different doses of the drug, either 5 milligrams twice a day or 20 milligrams twice a day. And they will be on the treatment, on the study for 26 weeks. Now, at the end of the study, people have the opportunity, if they want, if they decide to do so, to participate in a so-called open-label extension. Open-label extension means that basically everybody will be on the active drug. In the case of this study, everybody will be taking 20 milligrams twice a day of the compound for 1 year. And we believe that this study is also important to provide us more information about the long-term safety of the compound, and also to show us whether the drug has additional benefits in terms of slowing down the course of the disease. At the end, you can imagine we will have the 6-month study, phase 2 study, the Amarilla study, and the 1-year open-label extension study. So that gives us a lot of information on how this compound behaves over the course of time. And the Amarilla study will look at different aspects of the disease, just because of what I just mentioned to you. We think that this drug can improve not only motor, but also cognition and behavior problems in HD. Therefore, we want to test that in the study. This jargon that I put here, primary objective, secondary objective, exploratory objectives, is a way of categorizing or of dividing the goals of the study, which is typically used in clinical trials. So first of all, we want to make sure the primary objective, so the main goal is to make sure that we're improving motor function. But also secondary objectives, we want to make sure that it's safe, the compound is safe, that also does something positive on chorea, and we also want to test how the providers, how the investigators feel about the changes in the participants. So are people getting better, clinically getting better, or not at the end of the study? And then we will use a set of so-called exploratory objectives. So these are actually we will use a measurements that are not fully validated, have not been completely accepted by the community in terms of their utility to measure, cognition, behavior, and motor function. And so that's why they're called exploratory. We don't know exactly whether these are the best measurements, but we want to use them to test that. We will also ask patients, participants, to undergo a brain MRI. There is a subgroup of participants who will be asked to undergo a brain MRI at the beginning and at the end of the study. And the purpose of the brain MRI is to see whether the compound does anything to the loss of tissue in the striatum that we were talking about before, of that purple area of the brain that we were describing before. And the idea is that, if there is actually any evidence that the tissue is not atrophying, is not dying as much as in the control group, then there may be an activity of the compound to slow down the progression of the disease. And this will be even more clear, as I was saying before, in the from the data in the open label extension. Because at that point, people will have been in the study at the end of the open label extension for up to 1.5 years. And that's basically enough time to see whether the compound can really slow down the progression of the disease. Another important aspect of the study is that of assessing the changes in functional abilities of the participants. So, as you all know, sometimes the scales and what the doctors do in the office don't always reflect how people with HD may feel and do during the day. And so the functional assessment, these tests that are done during the study, are primarily trying to tease that out. So are people getting better not only because the investigator think they're getting better, because the instruments are telling us they're getting better, but are they also getting better because they feel better? They're doing better in their daily activities. And so that's also an important component of this study. All these measurements that I just described, including the MRI, of course, will be continued in the open label extension for that, as I said, will last 12 months. This is basically a description, graphic description, of where we are with the centers. So we have, as I said, the Canadian centers, the U.S. centers, UK, Germany, and Poland. I need to thank actually George and the HDSA for including the information of the study on the HDSA website. And we have partnered for these with CHDI and Enroll HD, and more recently, which I haven't included in this slide, with the Huntington study group in the U.S. Now, I mentioned to you before that there was some news from a recently completed study done in Paris, in France. The study was a short study. It was only 28 days. So you can imagine that in a disease that progresses over 15, 20 years, 28 days is not enough to measure everything that we want to measure. But the study was primarily designed and run to test the safety in a small number of HD of people with HD. And eventually, we ended up having 19 people for in each arm. There were two arms, one that received placebo and the other one that received the highest dose that people are receiving in the Amarilla study, 20 milligrams twice a day. And the study showed that the compound appears to be quite safe and well tolerated, in line with what we already knew from previous studies that where this compound was tested in schizophrenia. Now, as an aside, the program in schizophrenia is no longer active, and just because the compound did not appear to be efficacious, did not appear in schizophrenia, did not appear to improve the symptoms in people with schizophrenia. But that doesn't have any repercussion on the potential efficacy of the compound in Huntington disease, because as you can imagine, these are two completely different disorders that, although may share some aspects of pathology in the brain, they have completely different causes, and so I would not basically be concerned about what happened in the schizophrenia program. What it's actually nice to see from the schizophrenia program is that overall the compound is safe and well tolerated. And again, we can't make any conclusion until we have completed the program, but it's an important piece of information to have in mind. Another thing that the small study in Paris showed is that the people who were treated with a compound appear to do better on a task that measures so-called goal-directed behavior. So measures how people respond to specific stimuli, to specific inputs. In this case, in the case of this task, to a money incentive. So people were asked to squeeze a grip dynamometer that measures strength as much as they could in response to a reward that was either money or a happy, sad, or emotional phase of start. And it turned out that the people who received the compound actually were able to squeeze the grip much stronger than the people who were not treated with the compound. Again, this is very preliminary. We cannot draw too many conclusions based on the small number and the short duration of treatment. But it is encouraging because goal-directed behavior, this type of response, relies on the same circuitry that I was illustrating at the beginning. So on the same corticostriatal pathway that is damaged in HD. So if anything, the result of this study supports the supports the test of this compound in a larger study, which we are currently conducting, the Amarilla study. So, so again, with some cautiousness, I think that that's actually the result of the French study can certainly give us more confidence that we can move ahead with the program. I think this is pretty much all I would like to share, and I can share with you right now. But I'm pretty sure that there may be some questions, and I'll be very happy to answer them. So I think we have plenty of time now for those.
Thanks so much, Ottavio. Yeah, so please do, if you have any questions, type them now. As I try to get my computer to work, I'll just ask a question, Ottavio. It comes up a lot from patients and families that call and inquire about clinical trials. Is there travel reimbursement for participants that are interested?
Yes, there is. Every study participant who needs to travel and requires lodging will be provided reimbursement. Absolutely.
Okay. And Another requirement we've seen often in HD clinical trials is that a caregiver or loved one should attend with them on all site visits. Is that a requirement for the Amarilla study?
It is not, but at the same time, the study is limited at this point to people who do not require a caregiver. So that's unfortunately the drawback to this.
Okay. First question here from the audience, Ottavio, is that do you anticipate that the PDE10 inhibitor will affect disease progression?
It is a possibility. So we have data from animals that show that the treatment with a compound, the effect of the treatment with the compound may outlast basically the duration of exposure. So I'll give you more details. So what happened is that some animals were actually treated for 4 months. Then the treatment was stopped, and the animals were tested between 2 days and 2 weeks later. And the animals that were treated with a compound were already doing better than the animals that were not treated. These are animals that these are basically animal models for HD. So although, again, that's still very preliminary, that makes us think that there is the possibility that the compound may slow down the progression of the disease. But we will not know it for sure until we have completed the current study, the Amarilla study, and its open-label extension.
Okay. Another question is, so you're testing the drug in early HD stage 1 and 2 patients. Do you feel that there could be benefit to people in more advanced stages of HD? And will you be doing later testing on HD patients in more advanced stages? So,
a limitation in a way of this type of treatment in later stages is due to the fact that eventually that area of the brain and the enzyme, the PDE10 enzyme in the area of the brain, disappears. So the reason that we are starting with people who are in the early stages of HD is primarily due to the fact that we still have enough enzyme, there is still enough enzyme in the brain to target. So we probably later on, I mean, it would be certainly justified to go with people in later stages. But unfortunately, at the beginning of testing this compound in people with Huntington disease, we need to be very cautious and go basically with the patients that we know have enough PDE10 left.
Mm-hmm. Here's a fun question, and just coming from the audience is, why is this study called the Amarilla study? Could you tell everybody where that name came from?
Yeah, no, absolutely. Actually, thanks for that question, because it's nice to hear the story. When a clinical study is prepared, there is always a brainstorming of the team. What do we need? How do we call this a study? And often you see these very complicated acronyms that groups come up with. This is actually the idea of the Amaryllis came to one of my colleagues, who's the clinical lead on the program, Marielle. The Amaryllis is the symbol of basically the I would say the fight against Huntington disease. It's the symbol adopted by the European Huntington Association. And the Amaryllis is a flower and symbolizes basically rebirth. And we thought that it would be a nice thing to have in the study. And it looks like it has been well received in Europe by the EHA, the European Huntington Association, as well.
That's great. Another question. You got a lot of questions, Ottavio, so I'm glad we have some time. So there's a lot of excitement about this study, which is fantastic. In the schizophrenia program that you alluded to, was there evidence of the Pfizer drug actually hitting its target in binding to PDE10?
Yes, we have actually more than that. We have run this must have been a very well-informed person. we have actually done a study to look specifically at the binding of PD-10 in the brain, and we basically saw from this study that that's the case. PD-10, a compound binds to PD-10 in the brain, and does it pretty well. So yes, it basically the compound reaches its target in the brain.
Yeah. Another one relating to how you define, what are the symptoms, or how do you define stage 1 and stage 2 for participants in this study?
Yes, that's actually based on the total functional capacity score that investigators take at the beginning of the study, and it's a component of a larger scale called the Unified Huntington Disease Research Scale. Mm-hmm. And the purpose of this scale is to basically measure how functional people are, and so the scale is divided in ranges. And we're basically picking only the top 2 ranges, so the HD1 and HD2 of the scale.
Okay. Couple questions from folks about the age requirements. So can you just briefly discuss why was the cutoff at 30 and 60, between 30 and 65 years of age for participating in this study?
Sure, that's a very good question. The reason was primarily to try to see how the drug behaves in adult individuals, in the individuals with the adult form of HD. So the initial idea was to limit to the adult HD because we know for sure that people in the early stages of HD who have the adult form have enough PD-10 in the brain, and we don't know for sure if that's the case of the juvenile form or the late onset form. There is to say also that the juvenile form of HD manifests in a slightly different way, and so running, including basically people with juvenile HD in the same study would have posed some challenges in terms of how measuring changes and over time in the same way. You can imagine that, for example, young people with juvenile HD have a higher rate of seizures as well, and other aspects of movement disorders that are not typically seen in older individuals. And so that would have created more difficulties in testing the effect of the compound at the same time in the same way across the 2 different populations. George, are you there?
Sorry, I was on mute. So next question for you, Ottavio, is, as you know, a lot of HD patients, or most HD patients, are on different medications to treat their symptoms. Do participants in our Amarilla have to get off their medications to participate in this trial?
Yeah, that's also a very good question. It depends. It depends on the medications. There are some medications that typically used that we allow, for example, antidepressants, neuroleptics or antipsychotics, some medications used for anxiety, for example. There are other medications that we typically exclude, but primarily because they may either interact with a compound or may pose a risk by themselves. So we can't have them in an investigational study like this one. But overall, the vast majority of medications typically used in HD are accepted.
Okay. And I guess so it's kind of a question kind of related to this. Does participation in the Amarilla study, would it affect a patient's ability to participate in other studies or trials?
It depends. If it's another investigational study, So another study where the person needs to receive a drug or an active treatment of any sort, even psychotherapy, let's say, then yes. If the other study is a so-called observational study, where basically the person needs only to be followed and monitored, like Enroll HD, for example, then there is no problem in participating in a study like that.
Another question, this is related to target engagement. This is coming from obviously another scientist that's participating, is are you demonstrating in this study, how will you be demonstrating that you've engaged PDE10 in this particular study? Is that being looked at to make sure?
Well, not in this particular study. So we are relying now on the information that we have from previous studies that show that we can engage the striatum and PD10 with this compound. So we thought it was not necessary to do the same in this study. So we know that the drug gets to the target at this point.
Mm-hmm. Great. Another question. I know you mentioned that the study is 26 weeks in duration. Could you comment on the number of visits within that 26 weeks that a patient and family can expect? And once they are at the clinic, you know, are we talking an hour visit, or is this a day-long visit to do all of the different studies? Yeah,
so unfortunately, it's not probably the shortest study in terms of visits that you can imagine, but for a good reason, because we want to obtain enough information, longitudinal, so over the course of the study, to be sure at the end that the results are credible, they're true. So we have actually a period, so-called period of titration at the beginning of the study, where participants are asked to come back after 1 week, 2 weeks, and 4 weeks. And the purpose of this titration phase is to make sure that the drug is well tolerated at the most important time, when you basically increase the dose of the drug. So we want to make sure that people are responding well and there is no problem. Afterwards, there are other visits that are scattered, they are more sparse across the study duration. So after the first month, people are asked to come back at 3 months, and then eventually at 6 months. I think I'm missing a visit. There are a total of 9 visits, basically, throughout the study. There is also a screening visit at the beginning and the baseline visit. Okay. But most of the visits are concentrated in the first month. Now, the 3 of those visits, so you know, the first visit before starting to take the study drug, and the 3-month visit and the 6-month visit may be a little bit longer. I would say people may expect to be at the site for 6 hours, sometimes even longer. It all depends on many things, on how well the visit can proceed, and the availability of the staff at the site at a specific time of the visit. But overall, I would say 6 hours. The other visits in between are usually shorter.
Okay. Great. Thank you, Ottavio. This is just another question from a family member regarding the age criteria. There's a loved one that's 66 or 67 years old. Are those numbers? Is there any wiggle room in that inclusionary criteria of 30 to 65 years old? If somebody was 66, or is it just a hard line?
Yeah, unfortunately, those are hard numbers. Yeah. And I know how it may feel, but once you have those numbers in a protocol, you really need to draw a line, and you cannot be too permissive. So yes, unfortunately, those are hard numbers.
Okay, thank you. Is there a couple more technical questions before I get to one that there's a question that many people have been asking, and we'll get to that at the very end. But are there differences in target engagement with this drug between schizophrenics and HD patients?
Target engagement. So I'm not sure that I fully understand. So in terms of target engagement, there is not a difference that I can think of between schizophrenia and HD. So the compound binds the PDE10 independently of the disease.
Mm-hmm.
What is different is the effect, the downstream effect of the compound. So as you can imagine, schizophrenia, it's not only due to corticostriatal dysfunction. There are many other parts of the brain that are responsible. And also people with schizophrenia have, often are also on different medications. So it's a completely different disease, often treated in a very different way. It's true that, both people with schizophrenia and Huntington may take antipsychotics, but the doses of antipsychotics are usually different, and the treatment is much more prolonged in people with schizophrenia. So all this to say that it's not the difference between schizophrenia and Huntington are not only related to the pathology, to underlying condition, but also to the treatment, and that makes basically the response to PDE10 inhibitor in the two conditions not comparable. So it's really, you could not predict how the compound you cannot predict how the compound will behave in people with HD based on what it did in people with schizophrenia.
Okay, thank you. Another question is that, do you have any data, I guess this would be probably preclinical, I don't imagine you have clinical data, to provide confidence that you'll be able to, with inhibiting PDE10A with your drug, that you'll actually be able to see volumetric changes using MRI as your readout?
Yeah, unfortunately, the animal models do not lend themselves to that sort of analysis. Yes, that's a very, very good question, but it's something that cannot be sorted out based on a morphometric analysis of the
A very common question here. I know I have one answer for this, and Octavio, I'll be interested in your answer as well. As folks want to know how to best get involved, where can they find the list of sites, potential coordinators of where they can get involved in this study if they're interested in. And hopefully everyone can see this slide. HDSA, if you haven't heard of it, we do have a new free online resource available to folks. This study is listed in the database as a potential option for folks. So you can go to hdsa.org or hdtrialfinder.org. You answer a short, set up a profile, answer a short questionnaire, and it'll match you to the clinical trial sites that are closest to you. And all the listings of all the actively recruiting Amarilla study sites should be in this HD Trial Finder database. So if you haven't seen it, I encourage you to take a look at it, and your local contact information should be there. But Octavio, I'm just speaking on behalf of me and HDSA. I don't know if you have any other suggestions for people if they're interested in participating. What should they do?
No, this is a great suggestion. Thank you, George. Well, what I would recommend, if anybody's interested in the study, they should reach out to their provider. We are connected with most of the largest HD centers in the country. I think this information also publicly available. If you go on clinicaltrials.gov, they should be updated. It's basically, this is a government, it's a government, NIH government website where you can find information about clinical trials. And so if you basically type a PD10 and Huntington on this website, clinicaltrials.gov, you should come up with a description of the study and the site. But again, asking your asking a provider will certainly be another good way of connecting with the centers.
Great, thanks, Ottavio. So yeah, thank you so much for presenting this really exciting study to us in the community. As I said, this is being recorded, and we will archive this onto the HDSA website as well as our YouTube channel, so you can share it amongst families and friends as you see fit. And if you have any other questions, feel free to write me here at HDSA. And if there are questions that we just inadvertently skipped over, which we're happy to try to answer them after this call. So thanks again, Ottavio, and for your participation. And please join us next month on December 9th at 12 o'clock for our next research webinar. Until then, thank you so much, and we'll talk to you soon. Bye-bye.
Thank you, George. Bye-bye.
Take care.